So it is an honor for us to present Dr. Michael Telias, who will tell us a lot about his exciting research. Thanks for joining us.
Thank you so much for the invitation. I'm going to be talking today about a few projects that are going on in my lab, but mostly the most important one, which is pathophysiological remodeling of the inner retina in diseases that cause photoreceptor degeneration. As you well know, people can go blind for many reasons. We focus specifically on diseases that cause photoreceptor degeneration, especially retinitis pigmentosa. And so this is an overview. Let's skip that. I'm going to go back to that in a moment, but these are the two most important things that are going on right now, and I want to focus on that. And if we have time, we can talk about other things later. You guys all know the structure of the retina. Of course, you have the photoreceptors on one side, on the posterior side, and the ganglion cell layer on the anterior side. And these cells here are the ones that form the optic nerve. So basically, whatever happens in the retina that affects the communication between the retina and the brain, these cells are the last station before the train leaves forever, right? And so we want to understand what happens to these cells and the inner retina as a whole in diseases that cause photoreceptor degeneration. And we do that in the lab by using two mice. One mouse is called the RD10 mouse. It is a slow and mild form of RP in which not all the photoreceptors are gone. There is actually a layer of cones that remain there for a long time. And it takes a while for the mouse to go blind. The second model is exactly the opposite. It's a very aggressive, very fast form of degeneration. It's called the RD1. These mice basically are born blind, if you will. They show full degeneration by P14 or even before IO. And what people have shown in the past is that as the photoreceptors die and after they're dead, there is a series of different changes that affect the inner retina that we collectively call remodeling, and they include many things like the creation of new dendrites, like gliosis, like oscillations and electrical coupling that are enhanced among those cells, among amacrine cells and bipolar cells. But two phenomena within the world of remodeling, specifically the hyperactivity of ganglion cells and the membrane hyperpermeability of ganglion cells, are the two that I'm going to focus on, and for reasons that will become clear in a moment. And so I just want to mention before I move on that this is not by any chance a product of the mutation that causes RP, because we have shown and others have shown that in mice where you induce retinal degeneration with light, or where you just create a local injury on the photoreceptors, and even in human samples with AMD and RP, you get remodeling, okay? So we are using mice with specific mutations, but the mutations are not the cause of the remodeling. The remodeling is downstream to the death of the photoreceptors, no matter how the photoreceptors die or how many of them die. Even if it's just 3% of the retina. In this paper, we showed that when you kill only 3% of the photoreceptors, you still get remodeling underneath that local injury. And this was done by implanting one of the implants that you work with, Palanker. And we came to our lab and just implanted a bunch of mice and rats. And then we looked at those retinas and we found that. So this is how remodeling looks like. One of the first things that you see as soon as the mouse starts to go blind and starts to lose photoreceptors is the hyperactivity and the membrane hyperpermeability. So these are results from Steven Stashev many years ago, where he performed MEA, multi-electrode array recordings of RD1 retinas, white retinas, and you can see very easily how the hyperactivity, the spontaneous hyperactivity of ganglion cells arises very early in the process, because this is a fast and aggressive mutation, and it remains for a long time in the mouse retina. And we showed, Richard's lab showed when I was a postdoc there, that in parallel to that, dyes that do not penetrate membranes do penetrate the membranes of ganglion cells in degenerated retinas. And this is due to the opening of a receptor, of a massive receptor called the P2XM receptor, which I will be focusing on in a moment. But other people have shown other things, like the lab of Thomas Carrier showed that specific circuits develop intrinsic oscillations and enhance electrical coupling between amacrine cells and the off ganglion cells, specifically when the target is an off ganglion cell. And the work of Brian Johnson, Robert Mark, for many, many years, this is just a review paper, they showed massive changes, morphological changes. So if you wait long enough, when the photoreceptors die, not only the communication between the cells is altered, not only you get the hyperactivity and the hypernormality, but you can see that the layers are disrupted. Okay, amacrine cells move around, ganglion cells grow new dendrites, eliminate dendrites that they had before, and the same happens to bipolar cells. And the retina eventually looks like a mess, but this is very late stage of the disease. So this is how it looks in our hands. You can see how the RD10 mouse slowly degenerates. So at the beginning, it has beautiful light responses on, off, on, off. And then those responses start to dwindle by P28 when the degeneration starts. And then by P60, at this light intensity, you don't see any more light responses, but there is massive buildup of background noise. This is the spontaneous activity that we are talking about. And in RD1, from the go, you have that very strong spontaneous activity, hyperactivity. And the reason that I'm focusing on this is because when we want to restore vision, as I said before, the ganglion cells are the last train, are the last stop for the train before it leaves, right? So whether you're using a retinal prosthetics or we're using a stem cell therapy, you have to go through them. And this buildup of noise will basically reduce your signal, right? It's a classical signal to noise ratio problem where whatever signal you want to pass through these cells is going to have to overcome this noise. And so our question is, what is the source of this noise and can we block it? And so we showed that through complicated the series of steps, we arrived at the hypothesis that what is happening is that as the photoreceptors die, and it doesn't have to be the case that they're all dead, even at partial degeneration, you have massive influx of retinoids leaving the layer of the photoreceptors, who are dying right now. And those retinoids start to invade, to penetrate into the inner retina. And when they do that, they have no other option but to eventually get converted into retinoic acid, which is the end product of retinoids from vitamin A. Retinoic acid is a key morphogen in development, but it's also a key signaling molecule in adult life. And it has a receptor, a series of receptors, RAR receptors, retinoic acid receptor, which are expressed in all cells in the body. And so as you go blind, this buildup of retinoic acid activation in the ganglion cells causes the noise and causes the light responses to be swallowed in that noise. But when we block RAR, we basically reduce the noise and allow those signals to emerge from within the noise. And so how did we do that? Well, one approach that we had is to take the receptor. The receptor is a nuclear receptor, and it binds to specific sites in the DNA, the promoter of genes called RARES, retinoic acid response elements. And they just sit there waiting for retinoic acid to activate them. In the absence of retinoic acid, the receptor is always inhibited by a co-repressor. So there is a drug that is an agonist of the co-repressor. So it's going to be blocking retinoic acid retinoic acid receptor all the time, even if the ligand is still present. And when we do that and we intravitreally deliver this drug, BMS 493, into the retinas of RD1 mice, you can see for yourself how we, of course, this is all quantified in the paper. I'm just going a little fast here. We can reduce the noise significantly in the mouse with total photoreceptor loss.
And this takes how long does it?
3 days. We didn't see any effect after 24 hours. We started seeing an effect after 3 days, and we saw some effect even until 7 days, but not after 10 or 14. Those were the time points at which. Yeah.
And do you have any way to reduce this noise more immediately?
No, not that I am aware of. No, that's a good question. Now, if we do the same exercise, but now we use a mid-degeneration RD10 mouse with photoreceptors, with residual light responses, we take one mouse, we inject one eye with vehicle, one eye with the drug. Both eyes, both retinas are then subjected to the same kind of stimulation, the same light intensity, the same duration. You can see how a signal emerges in the treated eye, but is not present in the vehicle control. And these experiments were done double blind. No pun intended. And indeed, we saw in 5 out of 5 mice that every time the injected eye responded better to light than the uninjected eye, the vehicle injected. So that was great, but BMS 493 is an experimental drug. It's pretty toxic. You inject it in the eye, it stays there for only a few days, and you're done. People don't go blind overnight, okay? The RD10 mouse represents an RP person that can go blind for maybe 10, 20, 30 years of their life. So we looked for a different approach. And this is where we came across this drug called disulfiram, which is a non-specific blocker of ALDHs. And it also happens to be that it has high affinity specifically for the LDH that is expressed in the retina, RALDH, retinal aldehyde dehydrogenase. And the good thing about disulfiram is that you can give it systemically. And it goes through the gut, into the bloodstream, into the brain, and into the retina. And we showed that it really crosses the blood-brain barrier. And to make a very long story short, when we do that, we can take mice. This was done with Michael Telias from Santa Barbara, who's an expert in two-photon in vivo imaging in the brain. So we take RD10 mice at a very young age before the degeneration starts, and we give them 14 days of control food or food including disulfiram. Then we made a surgery where we inject a virus that delivers the calcium indicator GCAM6S and create a cranial window. And then we can in vivo stimulate the mouse and see the responses. And there were a bunch of different stimulations that they did, but one of the most pretty interesting one is this natural image simulator, if you will, which is just 30 seconds of the beginning of a movie in black and white. And so this movie has a sequence of black and white dots that create a sequence of activation in the brain that is known and has been already decoded in wild-type mice. And what they found in that piece of that work that we did together is that when you try to understand how well V1 in terms of GCAM responses, of calcium responses, is decoding these images and how close it is to the wild type, the disulfiram treated mice look much, much closer to real decoding than the untreated mice. Of course, if you compare it to a wild type mouse, the wild type mouse looks a lot better because they have intact photoreceptors, obviously, and they did also orientation tuning and contrast sensitivity and other things. This wasn't enough because maybe the brain responses are great and this decoding is awesome, but what if it's not enough for the mouse to actually see something, right? So we wanted to ask the question, are these mice now under disulfiram actually seeing better? So for that, we built a system that is based on a previous work many, many years ago by APU, where the mouse is running on a wheel that focuses the attention of the mouse on a screen right in front of them. And the screen is going to show a specific image that the mouse gets conditioned to react to by getting to the poke area, poking the nose in a specific time window and receiving a little drop of sugar water that mice really, really like. They like it more than cocaine. And so you can see here in the Hawaiian RD10 mouse at the beginning of degeneration that has been trained. Right? So it's running, it's looking for the water, it's looking to the screen, and the image appears, it leaves, immediately the wheel gets to the poke area, gets the reward, and then it starts again. When the same mouse goes old, and you can do this for many, many weeks, the mice don't forget about this task. The conditioning is just embedded in their brains. We show that with wild-type mice. The old mice are just too blind, and they just keep running. And they skip the image. And then what you can do is you can take that image and start playing with it and changing its contrast, its frequency, and whatever you like. And so when we did that in mice with disulfiram or without disulfiram, we see something pretty amazing. And this is the test at an early stage of degeneration where mice still see pretty good. Of course, this contrast sensitivity curve doesn't look very good when you compare it to a wild type. But the important thing from our perspective is that for the mice with disulfiram, we were able to pretty much preserve that contrast sensitivity. While the mice without disulfiram are functionally blind. Now, let me be clear here, and this was a question that we talked about before. We are not rescuing the photoreceptors. When we do ERG, the A wave and the B wave look the same. Okay, so this is just blocking the hyperactivity that is not allowing, in the case of the control mice, not allowing those signals that are still there to reach all the way to the brain. And in the presence of the disulfiram, by blocking the hyperactivity, we actually allow those signals to move forward and reach all the way. So disulfiram is a very old drug. It was discovered in factories more than a century ago when they were making tires for cars. And it was a byproduct of the process of making the tire material. And people just started puking whenever they drank alcohol. And so someone thought that this would be a very good deterrent against alcohol drinking, especially binge drinking. So people that suffer from alcohol abuse disorder, they get this drug for a few months, and that deters them from drinking. It's a non-specific blocker, it's FDA approved, it's already available, and it has known toxicology, pharmacodynamics, and pharmacokinetics. And as I said, it just blocks that pathway that prevents the breakdown of alcohol, which for us is great because it also prevents the synthesis of retinoic acid. So based on this idea, we have now 2 proof of concept clinical studies with disulfiram in patients with photoreceptor loss, whether it's IRDs, inherited retinal degenerations, or macular degeneration. One is going on in our institution at Rochester with Alex Levine, and the other one in Seattle with Rostam Gelder. So this looks very promising. This drug is cheap, it's accessible. If you don't drink alcohol, everything is fine, as long as you are not exposed to alcohol. But we don't know exactly what's going to happen, right? I mean, maybe 3 months of treatment with disulfiram for alcohol abuse disorder is not the same as 10 years of treatment with disulfiram for people with retinitis pigmentosa. And of course, we don't know if at those time periods we're going to get some chronic toxicity, accidental alcohol toxicity, and maybe one other thing that we are worried about is that the retinoic acid signaling system is known for engaging in feedback loops. So sometimes when you block something, something else goes up, right, to compensate for that. So maybe if you give someone disulfiram for 10 years, they will just, the RALVH will go up, or another member of the ALDH family will start synthesizing more retinoic acid. So it's not a perfect solution, but it's close to what we want.
Have you tried to see what happens when you drop off the drug?
When you what?
When you drop off the drug.
Oh, no. The time window to work with these mice is pretty short. It's about, maybe 30 days. So we wanted to do the reversal study that you suggest, but we never got. Yeah. And if you dark clear the RB10, you get an extra like 5 years. 5 days in the process. Like you delay it for like 5 more days, but it's not sufficient. Yeah. So I showed you that the activation of retinoic acid receptor in ganglion cells, maybe in other cells too, causes this hyperactivity. But how does it do it? Retinoic acid receptor is a transcription factor in the nucleus. It activates many genes. I mean, there can be so many other things downstream to it, right? But I also mentioned at the beginning that another phenomenon is the membrane hyperpermeability that you can see in the ganglion cells, and you can see their axons here. And in that case, we already knew that who's in charge of that, responsible for that, is this little guy, P2X7, not that little actually. Is an ATP receptor in the membrane of many neurons, not all of them, but also a lot of glia cells that express P2X7. And when this channel gets activated, it has a slow kinetic flow of sodium and calcium and potassium, but it opens a really large pore that allows these guys to flow in, and also cationic selectivity. So everything that is charged positive will go through that. And so we asked the question whether it can be the case that P2X7 is actually downstream to retinoic acid. And in the first paper in Neuron, we showed that when we block retinoic acid, we block the hyperpermeability, that was shown to be dependent on P2X7. So that was a first indication of that phenomenon. And here, a new study that we are hopefully about to submit very soon. We showed that actually my postdoc Leonor, who's also from Israel, she's showing that the P2X7 gene, both in mice and in humans, has a lot of rare sites in the promoter. Remember, these are the sites that retinoic acid receptor likes to bind to and activate whatever is downstream to them. And we also showed that if you just take a white-type retina and RD1 fully degenerated and you stain for P2X7, you see an increased staining of P2X7 in the inner retina of the RD1 mouse as compared to the one. These are non-specific bands of the secondary antibody that we show in a control experiment. So what we did is we found a mouse that is P2X7 knockout, and we crossbred it with an RD1. And now we have a mouse that is a mutant, double mutant. It's both blind, but also doesn't express P2X7 at all. You can see here in the RT-PCR that the RNA for P2X7 is gone. And then what we did is we took these mice, the RD1 mouse that goes blind, and we injected one eye with PBS, the vehicle control, and one eye with BMS 493, the drug that blocks retinoic acid. And when we lyse the tissue and we do Western blot for P2X7, you can see that the amount of P2X7, the amount of protein of P2X7 goes down, and it's completely gone in the knockout. But another thing that is interesting is that even in the white cell, so in the absence of disease, P2X7 is almost not expressed in the wild type mouse. And that is important for us because retinoic acid receptor is expressed in a wild type mouse. It just goes up with degeneration, but it's there in a wild type, in healthy tissue too. Here it seems to be that in a retinal lysate from a mouse, you don't see a lot of P2X7 at all as compared to the knockout. So now that we have established that retinoic acid receptor is upstream to P2X7, and P2X7 seems to be the effector of retinoic acid receptor, we asked whether P2X7 is necessary and sufficient to create that RAR-dependent RGC hyperactivity. And here we perform MEA recordings on the RD1 and the double mutant. And you can see that whether we block all the synapses onto the RGCs or not, it doesn't really matter. Only knocking out P2X7 reduces the noise. Perfusing the retina acutely with a blocker of P2X7 called TNT-ATP doesn't do that. So it's not something that you can block, in 15 minutes or in an hour with a drug. It's something that needs a signaling cascade that goes downstream to P2X7 and needs a continuous inhibition or a blockade. We did the opposite experiment. So we took a wild-type mouse and we overexpressed P2X7 in their ganglion cells. And you can see that the average firing in the dark spontaneous activity of a wild-type retina is pretty low. It goes down when you block synapses, but in both cases, it goes up when you overexpress P2X7. And so Leonor said, okay, let's take these mice and let's do an RNA-seq and try to understand what's different with them, right? So when I take an RD1, I compare it to another RD1, but one of them doesn't have P2X7, do genes go up or go down? And she found that most genes actually go down. And among those that went down significantly were a bunch of genes that are all involved in gated channel activity, calcium channel activity, calcium release, and other families. And what caught our eye was this, specifically the HCN1. So HCNs in general, are hyperpolarization cyclic nucleotide gated channels. These channels activate in hyperpolarization and cause depolarization by bringing in sodium and potassium, but mostly so. And so HCN1 is also very well known because of its role due to its role in cardiac physiology. So in cardiac physiology, when you need rhythmic activation, right? Every time there is depolarization, hyperpolarization, that activates HCN1, brings you back to depolarization, and that's how you get a rhythm. And but what does it do in the retina? It's not very well known, especially in ganglion cells. Remember, this RNA-seq is done both RD1, P2X7 knockout or not. So there are no photoreceptors here, to your question before. And so when we just started doing the hard experiment, which is the patch clamp experiment, we took retinas from wild type RD1 and the double mutant. We pole mount the retina and we just patch single cells one at a time. We fill them with dye and we look at there are no light responses in RD1, right? So the only way of knowing if they're on or off is by filling them with dye and seeing if the dendrites penetrate deep or not. If they go deep, they're off. If they go shallow, they're on. And that's how we divide them into on or off. And you can see that there's not much going on in the on cells, but there's a lot of going on in the off cells. There is a large conductance being activated at hyperpolarizatory voltages for the RD1, which gets completely reversed when we knock out P2X7, and that we identify as HCM1. We also have gene expression and immunohistochemistry assays showing the activation of HCM1 and how it goes down when you eliminate P2X7. So this goes well with what Ivan found many years before, almost 10 years ago, when he did the same, but in his case, he was using dyes and a mouse called the Taiwan YFP mouse that has individual ganglion cells labeled with YFP. And he showed that the penetration of the dyes, which is also P2X7 dependent, happens specifically or more, is prevalent more in the off RGCs. So this was really a very inducive experiment where we try to see if we can see in our electrophysiology something similar to what he saw in his imaging experiments. But this also opens the possibility that when you go blind and when you lose your photoreceptors, your ganglion cells are not affected equally. Off ganglion cells are being affected differently than on. Which means, how does that work when we want to restore vision, right? And also, even before we restore vision, how does that affect the brain? If one channel of information is differentially affected as opposed to others, then how does that work when we try to restore vision and when the brain is trying to make sense of the images that we are trying to restore? And so this is the inconvenient truth. The truth is that photoreceptor degeneration causes loss of, yes?
I want to ask with respect to human retina, because I'm not sure, you're saying they're on and they're off, but in human you sometimes have on center off surrounding the same cells. So would that be, these are separate three classes of cells, like human combine these two functions together in some of the ganglion cells?
Sure. So again, this is why this is why I'm calling them putative on and putative off, because the only way for me to know if they're on or off is where the dendrites go and not because of light responses.
I see.
Because the mice are completely blind.
Yeah, you don't know because you didn't examine them before they were.
I can. These are RD1, so they don't have photoreceptors from a very young age.
Whenever you say an on RTC, it means it's an on center of surround. It's the same.
Right.
Every ganglion cell has a center and surround, and they are off. They are completely.
There are several just blue and yellow that's maybe just on or off. But most of them are on and off together. But humans have
the on and off in color. It's a little bit different, but it can be in the center. It will be blue.
Okay.
In terms of illumination, it's always on center of surround or off center on surround.
So
it's a single cell, and it's the same.
Okay, so it's informal. Okay, thank you. What about on?
There are on-off cells. Yes, there are on-off cells, but they are the minority. And whenever we found those in our recordings, we discarded them because there were very few, and we didn't know.
Okay, thank you.
Really? In rats? Wow. Interesting talk. So this led us to the next stage of our work, in which we did the same idea. You don't change a horse that is winning, right? So we also looked for drugs that are out there and they can block P2X7. And the difference with retinoic acid receptor is that the P2X7 is on the membrane. It has a very large extracellular side, so it's more accessible to drugs and so on. And so there is a drug like that was developed already by Johnson & Johnson, and both Leonora and Emma, an undergrad in my lab, they started delivering this drug intraperitoneal to the mice based on the knowledge that this drug was already shown in humans, actually, to penetrate the blood-brain barrier. And we believe that whatever penetrates the blood-brain barrier, most of the times also penetrates the blood-retinal barrier. And so you can see that the hyperpermeability goes down. We block even 6 to 7 days after one single IP injection. We see the diloading of the ganglion cells, the hyperpermeability going down. And most interestingly, we see the hyperactivity going down. And the reason this looks different is that this is our new work with our new system. This is a 3-brain MEA chip with 4,096 electrodes. And if you look closely, you can see even the oscillations. Like the sparse of the oscillations in the individual trans. So this is something very, we're very optimistic about this and very happy with this result. Our next stage, of course, is we want to do two things. We want to bring this to behavioral assays and see if mice really see better due to this. It won't be in an RD1, it will be in an RD10, as I said before. And the second thing is to reformulate this drug in a way that we can use it as eye drop, as drops that go directly into the eye, penetrate the cornea, and get all the way into the retina and activate, inhibit P2X7 inside the retina. So the conclusions for this part are that degeneration-dependent inner retinal remodeling corrupts retinal responses and its communication with the brain. That RAR-dependent P2X7 activation causes RGC hyperactivity, is necessary and sufficient for RGC hyperactivity, and blocking RAR or blocking P2X7 rescues the phenotype and improves vision. And as I said before, this curious and interesting phenomenon in which this is affecting specifically of RGCs, and how will that affect visual processing when we try to restore vision. So if I have a few more minutes, do I have a few more minutes or not?
Yes, yes.
Yeah.
Okay, so a little bit of other.
You have 20 minutes, 15 minutes.
You go here.
So
As much as you
It's
can.
until night.
sure, we'll stay here until Pesach. So so we are working on other things in collaboration with other people, funded by other agencies and intramural funds. This is one project that we're doing with Sana Marcos and her students and postdocs, in which we are looking at the connection between retinoic acid and the development of myopia. And so what I showed you before is that we can take mice and block retinoic acid in a mouse that is going blind. What happens when you increase retinoic acid in a mouse that is perfectly healthy? What happens to that retina? And so it turns out that this causes remodeling, not only of the retina, but also of the whole eye. The eye starts to grow and the sclera becomes thin, and the collagen fibers in the sclera start to rearrange themselves. And so what these very talented students are doing, they are taking mice, feeding them with retinoic acid, running structural biometry on the refractive status and other parameters of the eye. And then at the end of the experiment, when they kill the mouse, they use second harmonic generation imaging of the sclera to see if the sclera is changing too. And I will not go into most of this, but the mice, C57 mice, completely healthy, no degeneration, no nothing, no mutation that we know of. When you feed them with sugar pellets that contain high amounts of retinoic acid for 2 weeks, they just become myopic with the diopter measurement going down and with the axial length going up. So the eye basically is growing and they become myopic. Mice naturally become hyperopic, by the way. That's how mice work. They just become hyperopic with time. And most interesting for us is that when we now, after doing these measurements, we kill the mouse and we bring the retinas to my lab and we put them on the LEA and we start shining light on them, what we see is a very interesting phenomenon. We see a crunching of the on versus off peak, firing peak. So the on response is always higher than the off in these mice, almost twice as much or more. And you can see here the beautiful responses on off, they're very consistent. When we feed them with retinoic acid, we see the emergence of the noise, as I showed you before, but for the RD1 mouse, which we showed that has increased retinoic acid, and we see this crunching of the on to off response in terms of their ratio. So now we are moving forward with this idea by trying to do the reverse experiment, which is to shine defocus light on normal retinas and see if they can cause the same phenomenon as crunching the peaks of the ratio of the two responses. Another thing that we are very interested in is the question of how is that the inner retina as a whole and the ganglion cells in particular survive the death of the photoreceptors. Photoreceptor layer in a mouse retina is like 70–80% of the cells. It's a massive loss. And these mice have a very aggressive mutation. So how is that, I don't want to use something very tragic here, but 70% of your family dies. How do you survive? How do you go on with life? Okay, something like that. Something very horrible. And so it turns out that actually, if you lose your photoreceptors, eventually, with a lag in time, you will eventually lose some of your ganglion cells. And so again, how do we restore vision if we lose also the ganglion cells, right? How do we connect to the brain? And so Alan is a master's student in my lab who's about to start his PhD. He takes the retinas and whole mounts the mouse retina. These have a little butterfly configuration, we call it. He stains the retinas with RBPMS, which is a marker for RGCs, and then he starts counting the amount of RBPMS stained cells from the center, from the optic disc, all the way to the periphery. And you can see that at all ages, even at young ages, you can see a slight difference that is not statistically significant. But at P300, you see a very, very steep drop, almost half the loss of ganglion cells in these mice. And so we are trying to move forward, not only just counting them, but also trying to classify them into groups by sheer anatomy, size of cells, alpha cells versus non-alpha cells, and so on. But we wanted to ask, okay, if we see a drop that is about, let's say, about half, that means that the other half of the cells are still alive. So our question was not what is killing one half. Our question was what is keeping the other half alive. And so we wanted to look at a specific system, which is the system of neurotrophic factors. As you know, neurotrophic factors like BDNF, NGF, these are factors that are secreted by neurons to promote neuronal growth during embryonic during embryogenesis, but also in adult life, they maintain cells and they create stem cells that repopulate the brain and so on. And so what we did is we did two different assays. One is a gene expression assay where we just lysate the retinas, but we're comparing a wild type to an RD1. So here you have photoreceptors, here you don't. And in that case, what happens is that if you just normalize to GAPDH or any housekeeping gene, you don't get any meaningful results. So instead of that, we normalize to a marker for ganglion cells, which is RBPMS. And in that case, specifically, we see that TRK-B, which is the receptor for BDNF, is upregulated at very young stages of the disease, a very young, early degeneration, early stage of the disease. And when we look at the immunolocalization of TRK-B in the retinas of wild-type mice versus RD1 mice, we can see a very strong upregulation of the protein in the inner retina, specifically in the ganglion cell layer. There is no colocalization with RBPMS here in these results, but we have now new ones where we colocalize with RBPMS, which is a technical issue with the antibodies. And so what we're doing now is we found out that there is a lab in Harvard, David Ginty's lab, who created a long time ago a mouse that has a cassette encoding eGFP under TrkB. So everywhere that TrkB is expressed, GFP is expressed too. And we crossbred it with wild-type mice, with normal photoreceptors. These mice themselves are albino. And with our RD1 mouse, the mouse with retinitis pigmentosa. And now we're looking directly at GFP instead of using antibodies. And you can see that in that case, we really see a very strong increase of TrkB dependent expression of GFP in the inner retina of these mice. There's a lot of TrkB, by the way, in the photoreceptors over there, but I will leave that for later in normal conditions. So now we are trying to perform the experiment in which we are going to block TrkB and see if that causes increased death of ganglion cells, and the opposite, when we agonize TrkB to see if that can preserve ganglion cells and give us more time of functional vision for these mice. And finally, the last part, the last project that we are, and I'm telling you all of this because I'm recruiting people. So if anyone is interested, we are trying to ask the question: okay, if you are losing your photoreceptors and your inner retina becomes hyperactive, does that translate into the brain? Is the brain seeing hyperactivity? Is the brain being affected by that? And so what we do, this is the neuropixel, by the way. This is the recording of neuropixel. You can see here 80 electrodes, 80 channels, sorry, out of the 300 and whatever that we have. This is again Leonore performing the experiment where we introduce one of these probes into V1, and then we shine light. In this case, these are moving bars, moving Gabor patches that are moving at the same speed all the time. We're changing the orientation, as you can see here, and we then can see how many cells are tuned to the stimulation and how many are not. In the RD1, of course, we see that the number of cells that are tuned to a specific orientation goes down, and that is to be expected. But then the question would be, what happens to this when we block hyperactivity in these mice? But it's going to be hard to do it with disulfiram. It's going to be hard to do it with intravitreal injections. If you do intravitreal injections, you can damage the eye itself, the cornea, the lens. So stimulating a mouse after that is not a good idea. So for that, we like to do a lot of transgenics in the lab. So we designed a new mouse, and this mouse has a dominant negative form of the retinoic acid receptor coupled to a TET-on cassette, and that is expressed under a RGC-specific retinal enhancer called the PLE260 PAC6 mini promoter. It's an artificial version of the promoter, of the PAC6 promoter, and it has a LOXP sites. So basically, once you cross these mice with these are all RD10 mice, once we cross them with RD10 mice expressing synapsin CRE, for example, or some other neural promoter with CRE, you get mice that express blue fluorescent protein in every ganglion cell, but only when you give these mice doxycycline, you start expressing the dominant negative form of the retinoic acid receptor, which then blocks the activation of retinoic acid receptor dependent genes. And the beauty about this is that doxycycline, it can be given by food or by water, and it's completely reversible. As opposed to disulfiram, you can reverse it within a few days. It doesn't take weeks or something like that. So we are very excited about this mouse, which is going to go into this maladaptive plasticity in the brain project. And we are advancing that research over there. And finally, the operant conditioning system that I showed you before is complicated. You have to train the mice. Some mice don't learn it, and you need to discard it. Okay? And there are questions about motivation, right? The mouse is motivated to perform because it's hungry or thirsty or whatever. What about doing something that is innate to mice, that mice don't need to learn? A reflex that is always there no matter what you do and who the mouse is. And so there is a very nice experiment in which, classical experiment of the shuttle box, where you have a mouse that goes into a box that has two chambers. You shine light on one side, the mouse moves to the other. And there is a different one called the looming dot experiment. So the looming dot is a reflex that mice have to avoid predators, right? So from the perspective of the mouse, when the clear skies become dark, it's because a hawk or something is coming to hunt them. And so they either run away or they freeze. So we took a box like that and we project on the sides on screens a looming dot, and you can see that the mouse is just happy. It freezes, and then only when it's safe, it starts moving again. Okay? And this response happens 100% of the time, every time. The mouse cannot help itself. It just is inherent to its behavior. And that in that way, we can do a lot more experiments faster. So this is something else that we like to do in my lab. We collaborate with people in BME, biomedical engineering, to design these devices and to create the code that allows us to show these things to us. So future directions, when it comes to science, we want to understand how RAR-dependent myeloplasticity affects the retina and the brain. We want to understand how retinal ganglion cells survive, and even if they can be regenerated. We are now starting a small pilot project with a collaborator from Texas on retinal neurophysiology in mice with Alzheimer's disease. And of course, try to understand this connection between RAR and P2X7, whether it's only in the retina, elsewhere, whether it's relevant for diseases beyond retinal degeneration or not. And as I said before, we also are working on tool development, whether it's transgenic mice or a visually guided behavioral paradigms, especially optimized for mice. So that's it. This is the lab. These are my current people. These are some people that are not in the lab anymore, but they contributed. And these are our funding agencies and our website. Thank you. Thank you very much for having me. Thank you, Yossi.
שאלות? שאלות?
I have one question. Thank you very much for the talk. Mostly about the hyperactivity of the retina. I'm interested to ask you whether you think it's going